Hello everyone and welcome to the NPTEL course on pharmacognosy and phytochemistry. We are dwelling into the world of carbohydrates, and in this session we will dwell more deep into carbohydrates which are obtained from animal sources. Yes, animal sources, and these carbohydrates are honey and chitin and chitosan derivatives. So these two carbohydrates are obtained from animals, that is honey from the honeybees, as well as chitin and chitosan from most of your crustaceans. So let's learn what are these carbohydrates and understand them. So honey, oftenly referred to as mud, is a traditional preparation which has been used for thousands of years by humankind but is gifted to us by honeybees. So honey is a secretion which is stored in a honeycomb, which is been deposited there by the bees belonging to different bee families such as your Apis dorsata, Apis florea, Apis indica, Apis mellifica, in some cases Apis mellifera, and many members of the bee family, especially the Apidae family member. They all belong to the order Hymenoptera. Now, if you see bees, they are abundant in forests, but they are also harvested and dwelled in apiaries world over for collection of honey, which is prized. And the main sources or the main places from where we get our honey of commerce is Africa, India, Jamaica, Australia, California, Chile, Great Britain and New Zealand. Now in some cases you will also see that bees are dwelled into a special forest or special group of trees to get that characteristic unique honey obtained from those plants. So what is this and what are the contents of honey, how it is produced, let's understand. So honey bees collect nectar from the plants. As you must have seen, they have a organelle called as proboscis, which is like a straw which draws the nectar. Now if you see the nectar from the flowers in terms of its composition, it is mostly water, that is about 75% water, and 25% of it contains sugars, which is predominantly sucrose. Now once your honeybee consumes it, it goes into a special place called as honey sac, which is located in its abdomen, and there it is with the help of enzyme invertase, it's actually converted into honey. Now this nectar is actually the food for honeybee. So some part of it is utilized by the bee and the remaining part is something they deposit inside the honeycomb. So they keep on depositing it within the honeycomb. You must have seen the cells almost hexagonal of it. I have just kept an illustration here. So once the cell is full of this nectar, they seal it with the beeswax, and beeswax is also a wax secreted from their very own body. So with the help of their limbs, they seal it. Now before sealing it, one more process happens. The first process happens is in their stomach, where they convert your sucrose with invertase enzyme into invert sugars. The second process is when they secrete it, it is still very much rich in water. Now sugars that are rich in water are prone to microbial contamination, fermentation and so on. So here nature plays an interesting role. So the bees are said to fan the cells with their wings so much so that the water content of it almost reverses. So initially the water content was 75% and the sugars were 25%. Now you have your sugars which are 75% and your water content goes to as low as 25%. Now what happens then? So if you have a very high sugar content, they exhibit what is called as osmotic pressure, and due to their high osmotic pressure, your microbes cannot thrive in such an environment, and as a result your honey is self-preserved and it is considered to be one of the product which has absolutely endless, which can stay for ages together. So even in fact, there have been people who have discovered honey stored for thousands of years and not deteriorated. But yes, this honey, as you store over a period of time, tends to darken. Now, once your cells are sealed, this is something where your mankind intervenes. So, what is done is this honeycomb is being cut off and then it is allowed to drain. Now, in some cases, it is also centrifuged, broken and centrifuged, so that all the liquid, all the honey within that oozes out, and then whatever the contents or the solids are, they are superficially skimmed. So, this is what is called as natural honey or raw honey. Now, raw honey is rich in moisture, that is about, like we discussed, 14 to 24%. It's rich in dextrose, levulose, which is obtained from fructose, and they are the contents, or they are the ones which are highest in terms of amounts in your honey. They are called as the invert sugars. You also have little quantity of sucrose, dextrins, gums. Now, a honey has a typical pleasant odor. It has traces of essential oils because they are obtained from flower as a source. So, they do carry little bit of aroma. They have beeswax because they have been obtained from your honeycomb. Traces and quantities of pollen grains. So, when one is consuming raw honey, if you see raw honey, it's very translucent and turbid. This translucent and turbid appearance of honey is mainly due to first pollen grains, and the second reason for it is low water content. Because of its low water content or low moisture content, many of the sugars tend to crystallize in it. You might have even seen your raw honey, if you keep it in your cupboard for a long time, you might see some crystallization happen. So that is because the water content is really low. So this gives you turbidity to raw honey. It contains acids which are result of secretion of the bee. So you have your formic acid, acetic acid, succinic acid. Then you have your maltose, dextrin, some pigments obtained from flowers as well as vitamins. Now in your raw honey, even the enzymes are very active. So you have your diastase, invertase, inulase, whatever those digestive enzymes which were there in the gut of the bee, which is there in the honey sac, those are all secreted and are still viable within the honey, or especially the raw honey. So now what happens is, if you see your commercial samples of honey, you would say that we never see crystallization occurring there. Now what happens is, if you take your raw honey, the raw honey contains about 14 to 24% water and it has a good specific gravity, about 1.47. So it's very dense in nature and it is self-sustaining, like it has an endless shelf life. But in order to transport, in order to ensure uniformity, quality, and so that the sugars don't crystallize out, many of those commercial industries dilute the honey with water. So from specific gravity about 1.47, it is diluted to bring the specific gravity to about 1.35. Now when you bring down the gravity, you increase water, but at the same time you are going to decrease the sugar concentration. Now when there is a decrease in sugar concentration and there is more fluidity, again there is a good chance of fermentation. Now this fermentation and deterioration is being brought about by microorganisms as well as enzymes. So what is done, in order to take care of this enzymes, your honey is partly treated. Now how do you treat it? It's very similar to your pasteurization. Your honey is taken, it's diluted to bring about a specific gravity of 1.35. Once that is done, it is kept at 80 degrees Celsius for some time. Now if you keep it for very long time, you know sugars tend to caramelize. We don't want that to happen. So immediately after 80 degrees Celsius, it is cooled rapidly to your room temperature and stored. Now this treatment takes care of your enzymes and prevents fermentation of honey. But in the process, somewhere a compromise on the shelf life of honey is also being made. So if you see your marketed samples, most of the marketed samples will predict the shelf life of prepared honey, or what is called as industrially prepared honey. It is not artificial honey, it is just processed. So because of the processing conditions, now you have a shelf life which is generally given between 1 to 1.5 year, mostly 18 months time. So this honey is used as sweetening agent. Now there is a huge dispute whether it is good for health or not. Now if you're taking a diabetic case where you don't want to consume sugars, let us understand that because the invert sugars are already there in it, and that's a good high percentage, almost 75% of the composition of honey is because of sugars. It is a calorific sweetener. So it is going to give you calories and cannot be used for diabetic patients. But yes, I mean, if you compare it to sucrose, it has its little advantages in terms of other effects. Now this other effects include demulcent, especially when you have your throat irritation or throat disorders. We give honey because it has a nice soothing, it relieves your dryness, and it is also antimicrobial. Despite the dilution, it still remains a very high sugar containing, so it exhibits a high osmotic pressure. Your microbes cannot stand such a high osmotic pressure, and as a result they die off. So it's a very good antimicrobial agent, and that's where this can be said to almost be acting like a preservative. Now because it contains monosaccharides as compared to all the drugs we have studied previously, now monosaccharides are easily absorbable and easily bioavailable. So for infants or for elderly patients, or even for a convulsing patients, you will see that it becomes an easy food or easy source of energy. So honey is often given to young children as well as elderly for as a rich source of energy. But if you talk in terms of raw honey, because it has enzymes, because it has pollens, because it has other acids, it has a very strong antimicrobial and immunomodulatory activity as compared to your processed honey. Now because it's a animal product, it's priced, and you will see numerous adulterations happening where people create artificial syrup or add some sugars, and definitely it is contaminated, adulterated, or in some cases completely substituted. There is a simple test called as Fees test which can be done. So what you need to do is you just take your honey and then extract that honey with your petither. Now take this petither layer, and to that petither layer, if you just treat it with resorcinol and HCl, you might get a reddish coloration. Now in natural honeys, this coloration is transient, that is it will fade after some time. But if it is artificial honey, it stays for a longer period of time. Not only that, there are techniques nowadays which have been used to standardize honey. But this includes ratio of your sugars, especially sucrose, fructose. This also contains measuring your hydroxymethyl furfural, which is naturally present but more abundant in your artificial honey. It also involves measuring other percentages, specific gravity, aroma, presence of pollens, and many other tests have been devised to check the quality of honey. Now let's go to the second animal based product, and that's your chitin and chitosan. Now these are almost similar products. We'll see how. Now they have been obtained from, as you can see, crustaceans. They have been obtained from spiders such as tarantula. Now these are naturally occurring amine, that is your amino containing polysaccharide. So imagine your glucose, and if you attach an amine group to a glucose, and more specifically if you attach a acid amido group to glucose, that's where you get your chitin and chitosan derivatives. So these are nothing but your sugars or polysaccharide derivatives, wherein you have your chitin and chitosan coming out from these animals. Now there are numerous animals which can give you chitin and chitosan, like tarantula, crustaceans. You can obtain them from insects, worms. You can obtain them from the hyphal or the mycelial regions of certain fungus. Now they are huge molecular weight compounds, somewhere ranging between some million Daltons. So if you see chitosan is itself a high molecular weight compound, wherein the molecular weight ranges somewhere from 50 kilodalton to about 2000 kilodalton. Now what specifically are this? Now let's discuss little bit here first. So if you see this is what I was talking about, and this is your glucose. To the glucose you attach your amino group, and more specifically acetamino group. Now this you attach to another acetamino containing glucose by what are called as beta linkages. You get what is called as chitin. Now this chitin, if you selectively deacetylate, what is left back is just the amine groups. Those are called as chitosan. So it's not completely deacetylated. Some acetyl groups still remain, but the difference between chitin and chitosan is little deacetylated version of your chitin. So these are obtained from exoskeletons. Like I said, if you seen your insects during their growth, they undergo what is called as molting, where they lay their skin like the external coating, and that molting layer, you must have seen, it's whitish in color, transparent to almost translucent. In crustaceans it's not easily visible, because crustaceans, even if you see crabs, they might contain some pigments in it. But the insect exoskeletons or your fungal mycelia are very clear, transparent, translucent to white. In crustaceans they are embedded within the calcified layer. So what is done is, in food industry, as it is the crustacean meat is taken. So take the crustacean shells which have been discarded, and because it contains calcium you need to demineralize it. Now this demineralization is done with help of acids. Imagine an acid like hydrochloric acid or sulfuric acid. In most cases, let's take an example of hydrochloric acid. Suppose if you keep a crustacean shell in hydrochloric acid, this hydrochloric acid will react with the calcium there to form your calcium chloride. Now this calcium chloride is water soluble, so it gets dissolved. So over a period of time when you're digesting it, what you're going to get is a clear translucent shell which is demineralized. Now this demineralized shell gets a pliable, soft kind of consistency, but still contains one more component which is present in animal, and that is protein. Now generally the carbohydrates and proteins form a good association. The reason being, if you see carbohydrates, they have hydroxyl groups, whereas your proteins have amine groups, and then they tend to form interactions, especially due to forces of attraction. That can be detangled with by treating it with alkali such as sodium hydroxide. So the moment you treat it with sodium hydroxide, the associations are lost and your carbohydrates and proteins are separated. Now your carbohydrate is taken and it's depigmented. Like I said, one problem with the crustaceans is they have orange or kind of pigments present in them. Now these pigments are generally lipophilic pigments. So if you do take this crustacean pliable shell and break it into pieces and extract it in organic solvents, most of the pigments are lost. Okay. Or what you can do is, if you have a solution which contains this pigment, in some cases this depigmentation can be done by a simple process such as charcoal treatment. So the whole solution is passed through activated charcoal columns and you get a clear solution coming from it. Now this is the one which is rich in chitin. This can be concentrated, evaporated to get chitin flakes or sheets which can then be crushed. But in case if you want chitosan, you have to deacetylate it. Now deacetylation is done by enzyme called as chitin deacetylase, and in presence of more stronger concentration of sodium hydroxide, the deacetylation reaction is facilitated and you get a partially deacetylated product, and that is called as your chitosan. So this is the process in which you can get your chitin as well as chitosan. But apart from this, nowadays more greener enzymatic digestions have also been utilized for this purpose. So coming to the chemical composition, like I showed you, this is a glucose derivative containing acetamide group. Now this acetamide group, if you deacetylate, it is your chitosan. The linkages between two acetamide groups, or what you call it as D-glucosamine groups, are beta linkages. So in your chitin, it will be a homopolymer of beta 1,4 N-linked acetyl D-glucosamine residue, whereas chitosan, like I said, in some cases it is deacetylated, in some cases it is not. So you will get a mixture. So it will be a random distributed heteropolymer containing your beta 1,4 D-glucosamine, the deacetylated unit, and alternating or sometimes spaced between N-acetyl D-glucosamine derivatives, again linked by beta linkages. Now chitin, if you see in nature, occurs in different allomorph. Now this allomorphs are your alpha forms, beta forms, as well as your gamma forms. So what happens here is, if you see your alpha forms are in which the plates or the polymer sheets are anti-parallel to each other. So one sheet goes in this direction, the other sheet goes in this direction, and the alpha allomorph is the most commonly found one. The beta form is the one in which all the sheets are in a similar orientation. So this is alpha, this is your beta, and gamma is actually a mixture of A plus B. So you will find alpha, the chitin and you will find the chitin derivatives in alpha form, beta form, as well as gamma form, but mostly it will be found in the alpha form. Now if you see chitin, chitosan, they are yellowish to yellow white color powder, again depending upon the sources. They are odorless, the taste is slightly bland, and they are very resistant. They don't easily dissolve in water. So even in organic solvents, so creating a solution of chitin, chitosan, it's like, imagine your animal exoskeleton, you're trying to dissolve, it is that resistant. So how do you dissolve this? It is said that you might try to dissolve chitin and chitosan. Chitin is very difficult to dissolve because we saw it is still has an acetylated group. Chitosan is little simpler to dissolve, and it has amine groups which are exposed after the deacetylation happen. So this amine groups can then be created into salts by giving or treating it with acid. So instead of water, if you give something acidic or use something acidic, your chitosan dissolves it. So in your formulations or in your preparations, when you are trying to dissolve chitosan, generally an acidic buffer or acid such as acetic acid is used to facilitate the dissolution. So once it is dissolved, it will form a nice viscous gelatinous polymer solution, generally preferred or generally solubilized at acidic pH. You can check your chitosan is genuine or not by few chemical tests. You just treat it with iodine and then you just add 10% sulfuric acid or just drop to it. You will see that it gives you a deep violet coloration. If you treat your chitosan with nitric acid, it easily form nitrates, and this chitosan nitrates crystallize form or almost yellowish color crystals. You will see it under the microscope. Now this crystals, if you see you under the polarized light with using Nicol prisms, you might even see that cross wire like appearance on the nitrate crystals of chitosan. It has numerous applications. Now chitosan is something which is obtained from animal sources. It is a glucose derivative and hence very biocompatible. It is resistant, it doesn't dissolve easily in water, and that's an advantage. So if you have a wound healing application being done, it is going to not be soluble easily in your exudations, as well as even if you put some water or by mistakenly you pour water on your wound, it's not going to get drenched out very easily. So it's used in wound healing preparations to treat cuts, burns. It has a good property. It is studied extensively and has been known as a bacteriostatic. immunologic, it has anti-tumoral, hemostatic as well as anticoagulant activities. It is used to prepare artificial skin or even corneal bandages. Like I said, it's not much affected by water, and compared to other sugars, it is more robust and biocompatible. So it's used as a suture thread, as well as, if you want to do some implants or some gum cicatrization or in bone repair and in dental surgery, this is where your chitin and chitosan derivatives are abundantly used. It is used in textile industry for sizing rayon, cotton, wool and even synthetic fibers. It has a good bioadhesivity as well as adhesivity to glass and plastics. One interesting application is in the sewage water or effluent water treatment. So it is known to separate organic compounds, it is known to chelate or take care of heavy metals, and even for the pesticides such as DDT or PCBs, that is your polychlorobenzene derivatives, it has been known to kind of contain in it and then precipitate or settle down so that your water gets very clear. So here are few references if you want more reading into this topic, and thank you everyone for your patient listening.